So nervous. All right, we're live. Hello, everyone. My name is Susanna Harris, and thank you so much for tuning in to my public PhD defense. It's kind of a weird situation, and I know a lot of people are in the same thing. So first of all, I am technically a PhD already, and the deal with this is that my department, microbiology and immunology at UNC Chapel Hill, we do a private defense first where we pass or fail, and I got through that a couple weeks ago. And then we do this public defense where it's sort of like a celebration. And I just wanted a chance to talk to my friends and family about my science one last time. So this is that. Again, feel free to hop into the chat and ask questions. We will be able to answer those hopefully at the end, but we'll see how this goes. I'm really excited to get to talk to you all about my research. And thank you again so much. All right, let's pop over here so you can actually see my slides. This is the title, kind of my dissertation, but really all that I was focused on was building communities, specifically of bacteria, to help increase bacterial colonization of plants. And my whole PhD was mostly in bacteria, but I do talk a lot about plants. So the most important thing for me about this is actually, why do we even bother doing this research? The government gives us money, and we have to figure out kind of what we can do with it to benefit the world. So my project specifically, pop over here, was I wanted to figure out how to address this problem, or I wanted to help address this problem. Major problem that we are facing, in addition to everything else, is that we are starting to face the fact that we are not going to be able to produce enough crops to feed the world. And in fact, it's projected that by about 2050, we're going to have this huge deficit between the number of crops we can actually make and we can give to people and the number that we need to feed the world. So how do we start thinking about doing this? One of the ways we can think about doing this is actually increasing the crop production on its own. So what we need to do is we need to make more plants. But here's the question, can we do that without making and using more chemicals? We know that those are extremely environmentally costly. We know that it's not something that we really want to be doing. And there's a lot of pushback against using GMO, even though I'm a huge proponent, a huge fan of GMO organisms. In fact, most of the bacteria I work with are genetically modified organisms because I've gotten to play with their genomes. But we have to start thinking about, can we take what we know from the natural world, what's already happening all around us, and can we use that to help keep plants healthy because they've somehow figured out through millions of years of evolution how to do that themselves. And we're just going to pull up a little bit of a video. I'm a little biased because I did help make this video from the Moorhead Planetarium and Science Center, but I really liked how this is showing that while we're looking at the top of the plants, we actually need to go below the surface. We need to understand what's happening down there in the soil. This is where plants take up all of their nutrients, mostly. This is where they interact with the soil, with the different water, all those things. But as you can see kind of floating through the screen right now, the other thing that plants do down here is they interact with a bunch of microbes. Like I said, I'm mostly talking about bacteria, and in my opinion, bacteria are one of the most important things because this little teaspoon represents, in 1 teaspoon of soil, there can be 1 billion bacteria. They're everywhere, and they do so many things for the plant. And one of the ideas that scientists have is to leverage these bacteria and use what they're already doing with the plant to help grow more plants. And this idea is that some bacteria will help plants grow, and these are called plant growth promoting bacteria. I'll be referring to them as PGPB throughout the talk. And in some way, these bacteria will help the plants to grow. They might increase the plant growth itself, they might increase the crop yield, or they can increase the development. They can even fight off different diseases that the plant might be facing. So what we're seeing over here is just this general idea that there's some sort of bacteria in healthy fields that are keeping these plants healthy, and that maybe we could take these bacteria or these other microbes and transplant them into fields that aren't doing so well in terms of making more plants. The bacterium that I work on is Bacillus subtilis, but a bunch of different species of bacilli can promote plant growth, and we're really interested in these bacilli for a few reasons. One, they already live in the soil, they're already found with plants. It's not something that we're adding in brand new. We don't know what's going to happen with these plants. Another really big thing is they form what are called hardy spores. So this is why we're focusing on bacilli, at least in my research, is that we know we can take these bacteria, and they will stay alive, they will stay moving, not moving, but they will stay alive, and we can then allow them to grow and keep moving, but they will stay in trucks and in transport. And this is a really pretty picture that was taken by someone in my lab, Sarah Yannerell, looking at what do these bacilli actually look like in their natural form. And I love biology, I love microbiology. I'm going to really miss this because I think this is one of the most beautiful pictures that I get to show. So what can they actually do, though? What's impressive about these bacilli? And what we know is that they can actually help plants grow and produce more crops. But a really cool thing is that they will inhibit or stop the growth of different pathogens. In this case, we're looking at this pathogen, Ralstonia solanacearum. And these are tomato plants. And if you've ever grown tomato plants, they probably shouldn't be looking like this. They look pretty dead and dry. And so in this case, the scientists added Ralstonia solanacearum, and then instead of anything else, they just added water, and you see some dead plants. Versus over here, if they added Bacillus subtilis, again, the bacterium that I'm using in my research, they can actually prevent these plants from dying because the bacillus inhibits the growth of Ralstonia. This is pretty cool, right? Like, why don't we just do this all the time? Why isn't this the main thing that we do? And it's something that happens a lot. It's already been sold. It happens all the time. You might have heard of probiotics. This is the idea that some bacteria will benefit the host, and we can sell those for the soil, and a lot of times it will help the plants grow. One of the biggest problems, though, is that it's not just a one and done thing. You know, we put this living organism out in the field with the plant, and not surprisingly, it doesn't behave exactly how we would like it to as it does in the laboratory. This is an example of another bacillus, and just what you're seeing here, and again, if there's a little citation underneath, that means that this is not my data. This is something that was done by other labs. One of the coolest things about science is we learn from each other, and so this paper showed where these scientists took FCB42, which is a different species of bacillus, and they put this on lettuce plants, and then they planted it in a field, and they were looking at, does this specific bacteria show up in the field over time? And what you can see is after about 2 to 4 weeks of planting, so as the lettuce is growing, they find fewer and fewer numbers of these bacteria. And what's important here is they can look at the genetics, looking for the genetic pieces of the bacteria in the soil, or they can look directly for the bacterium itself. So, again, what's the problem? The problem is that we have to keep reapplying this, and this makes it not so attractive to agricultural work. If we could use a chemical that works for twice as long or better, why would we not use that? My research was focused on this main question of, can we help these bacilli, these different species of bacillus, stay with the plant? Can we do that for longer, and potentially would that help the plant grow? I was really excited about this question, though, which is, can we use other bacteria to do that? Like I showed in that really cool video, what we know is there's a ton of other bacteria all over the plant, and they interact with each other. There's so many amazing papers out there that talk about how different bacteria will kind of communicate with chemicals, and it causes their neighbors to do totally different things. And it's definitely within the realm of possibilities. This has been shown in other systems that maybe having a friend or, a couple other friends would help bacilli stay with the plant for longer. Just because I think it's necessary, here is the basic background of my dissertation project. We always have to have a hypothesis, and again, like I said, because we know different bacteria interact and change each other's behaviors, my hypothesis was that we could find other soil-dwelling bacteria that might cause bacillus to attach to the plant more or stay with the plant more. And the aims of my project were to, one, I had to develop an experimental system. It wasn't enough for me to just look at how many plants get there, but I also had to understand whether or not they were going to stay on the plant over time. Kind of tricky, and I didn't really want to spend entire growing seasons doing this. Then once I had the experimental system up and running, I had to screen for co-inoculants. Again, these are kind of the helper bacteria. These are the other bacteria that are going to go along with bacillus, and look at whether or not they would increase the survival and the maintenance of bacillus on the plant root. And finally, because this overall point is, can we take something like this into the real world? Can we use this to benefit people and the taxpayers that are actually paying for this research and this opportunity? We wanted to know if these co-inoculants that we found to help Bacillus subtilis would actually be useful for other species of bacilli that are maybe more often used in these beneficial agricultural products. So I wanted to start here with my first aim, which was to develop this experimental system. And what we have here is, this was a paper that we published a year or two ago through Jove. And the basic outline is down here, but Jove is really cool because you get to make these videos actually explaining your science. This is not a plug for Jove. I just had a really great time making these videos, and I think it's a lot more helpful. So generally what I would do is I would grow these Arabidopsis thaliana seeds. It's sort of the lab rat version of plants. We know a lot about it. I would grow them on a mesh disc. And I would take these little seedlings, and after 8 to 10 days, these seeds would now be tiny little plants, and I could pull them directly out of the agar, which is what you're going to see in a second. So I pulled them out of the agar, and then I could put them in the liquid. And what you saw just a second ago is that I was scraping up bacteria and putting them into the liquid as well. What was really important here is, of course, this is our colonization step. This is, step 2 that you can see down here in our little schematic. And this was where the bacteria are going to colonize. This is where the bacteria are originally going to stick onto the root. And we know this can work really well because the bacteria in this case are in a liquid medium that has a lot of nutrients for them. And they're super happy, they grow, they swim around, and eventually they find the root and they stick to it. Bacillus loves this. Then what you just saw is that I put this in an incubator and I let it sit overnight. And here, the step that was important, the piece that was sort of new in my system, is that then I filled up a new 24-roll plate and added in this maintenance liquid. What I would do then is I would take out these little seedlings, and I really enjoyed this part. Take out these seedlings. This is a rinse version, but then I will transfer them into another new plate and allow them to grow again overnight in that incubator and see after that time how many bacteria are still stuck with the root. And this is the maintenance step down here. It'll probably be a little slow, but basically what happens then is that after that incubator step happens, I can pull out these seedlings and do a whole bunch of different things where I can either grind them up. Actually, I blow them up with sound. It's called sonication. And it's really fun and really noisy. Imagine if you were watching Harry Potter, that 4th movie, and the egg opens up and it starts screaming. That's exactly the same sound that I got to hear for years on end. So after pulling these seedlings out, I can do that and I can put them on plates. And this is called counting for CFU. That's colony forming units. I'm bringing this up because I'm going to talk about it more. But this basically tells us how many bacteria are on each of the plants. Instead of blowing them up, I can take these plants. Again, I can't blow them up and then look at them, but I can take these plants out and put them on a microscope slide and look at them directly. So that gives you a pretty good idea of what I'm doing. What am I even looking at? I really hate when you're watching a science presentation and you're seeing a bunch of graphs and you don't know what they are. So this is one of the most common graphs I use. There's 2 graphs that you're going to see. And basically what's happening over here is that on the left, this is that CFU, so the colony forming units are sort of measurement of how many bacteria are there. And then the different sample types will tell you maybe this is Bacillus on its own, maybe this is Bacillus with friend Y, with friend Z. And what you can see is how many bacilli are on that plant. So how many bacilli are on each of those seedlings. And what this looks like is maybe there's about 10 to the third, which is 1,000 bacteria per seedling, whereas in this sample there's about 100 times as many. And so what is kind of what I wanted to point out here is that what can happen is if it's different enough, there will be a little asterisk. And in this case, it just means that it's statistically significant. I'm not going to get into statistics, even though very recently I was asked. But basically, this is how scientists measure, is this different enough, or is this sort of a thing that we're seeing by chance given our experimental system? You might also see this graph just as a bar graph. The little lines in the middle are indicating what the average of all these samples are. So instead of having the line, it would just be a bar up and down. Usually I use this because it's really important to me to show all of the different data that you're actually looking at so that other scientists can look at it and say, I don't really agree. This other graph that I use a lot looks pretty similar, but the difference is that we're looking at relative CFU. So there's going to be this line at 1, which is our control sample. So the sample, in this case, it's going to be bacillus on its own, and then we're going to be looking at, in Y and Z, what numbers of Bacillus subtilis are there with the plant when these other bacilli are there. What that's going to look like is, again, if this is our bacillus, the average of the bacillus sample is going to be right at 1. That's how we normalize everything. That's our starting point. And so in this case, sample Y doesn't have a statistically different change. It might be a little bit lower because we can see that the average is lower, but over here, sample Z is high enough different. There's enough difference between these averages, between all the different samples from our baseline, that this would be statistically significant. We're always trying to get statistically significant data so that we can say more confidently that what we are looking at is actually different enough. So looking at one of these graphs with real data, when I went to test this system that I had built, that colonization and maintenance system, we first used this bacterium Pseudomonas simiae. It's another PGPB, different kind of bacteria, and it can stay with the plant it colonizes the plant really well. We've got 10 to the 5th, 10 to the 6th, and also here you can see across different days there is some variation. But what is cool about this is that on each of these different days, day 1, here, day 2, these different biological replicates that are done at totally different times, we do see that after 72 hours in that maintenance media, we don't see a decrease in the number of Pseudomonas that are surviving on the plant root. So this is, again, that idea of we're taking the plants out, we're smashing them up with sound, we're looking for how many bacteria, and what we see is that Pseudomonas simiae stays really well with the plant in our system. And this was exciting because it said we have a system where bacteria can maintain their colonization even in that maintenance media that does have a lower number of nutrients. What we can also do using that microscopy approach, if this is a seedling, when we pull it out, these little boxes down here indicate where we're taking these microscopy pictures. And in this case, Pseudomonas simiae is a really awesome bacterium, sometimes I say bug, and it glows green, or it fluorescence in the green channel. And here I have it false colored in green. And what you can see, hopefully, is that these different plants that have been colonized, here's the no bacterial control down here. This is showing that on our seedlings without the Pseudomonas, we don't see that bright green showing up on the plants, or if we just look at the green channel. But here, even using 2 different microscopes, we can clearly see that Pseudomonas is colonizing the surface of these plants. We also wanted to see if the system would work for natural isolates that are growing on the plants in the soil, and so we got a bunch of strains from the Dangle lab. That's Jeff Dangle here at UNC as well. And what we can see is that these different strains of bacteria, Nicotinovorans, all of these different lovely names, they will colonize at about those same levels, and they can maintain. And the reason that we were looking at these bacteria are not just because they're found in the soil, but we can put them on plates together and see that they're very different. And that allowed us to do something cool, which is to put them all together, either under colonization or during maintenance, and pick out the individual bacteria when they've all been together on the plate. So we can count how many of the Mycobacterium that we have, Curtobacterium, etc. Again, we saw that there really wasn't a difference between colonization and maintenance, or not really much of a difference when we had them all together. Nothing statistically significant. Great. The assay works. My next aim was to then screen or look for helper bacteria that could increase Bacillus subtilis maintenance. And so the reason we were looking at this is, again, we used our system, and so in this case, Bacillus subtilis is false colored blue. This is the root crown, this is the root tip, so if it's in the ground, this would be the surface up here, and false colored blue, we can see bacillus growing all over this plant, and over here in that CFU per seedling, we've got about 10 to the 4th, 10 to the 5th bacillus. There's a pretty good variation, and this is very normal. However, after we put this through our entire system, what we ended up seeing is that there weren't very many bacteria, there weren't very many bacillus on the root. You can see that after just 12 hours in our system, they fell off, which was exciting. This allowed us to probe for maintenance. And even on the root, you could barely find them. They looked really sad. We wanted to see if we could turn this whole thing around. So we were looking for Rhizosphere isolates. These are bacteria, again, that live on the plant root or in the soil around it. The rhizoplane is the surface of the plant root. And find ones that would support Bacillus subtilis staying with that plant. What we ended up finding is we took 96 isolates, all of these cool bacteria. We know all of their genomes. They're really fun to play with. These are from the Dangle lab again. And we looked at them for their ability to individually maintain. We found 51 of these 96 that could maintain in our system, sort of similar to Pseudomonas simiae as before. And 22 of them we found as possible helpers for Bacillus subtilis. And in a giant picture format, what this looks like here, this is a phylogenetic tree. All we're looking at here is how related these bacteria are to each other. And the little orange dots indicate which ones might have helped Bacillus, and the greens are those that were able to maintain colonization. There didn't seem to be much of a correlation between maintaining on their own and helping Bacillus, nor helping Bacillus and their relatedness as different bacteria. We took those 22, and like you saw with all the variants, there's a bunch of things going on. We screened all of these again many, many times. And the main thing to take away from this is that of these 22, 5 we found to be statistically significant. So this is just a twisted version of that second graph that I showed you, where in the middle here, this red line indicates 1. This is the average of Bacillus when it's on its own, versus all of these other dots, and it's important to keep this in mind, all of these other dots are not the other bacteria. Rather, these other dots are how many bacilli are in this sample. How many bacilli, when mixed with a different bacteria and added to the plant, how many of those come back off? And so 5 different strains were able to significantly increase how many bacilli stayed with the plant after our system. And 3 of those we chose because, as you can see, all of these different samples are above that red line. There's a pretty small variance compared to some of these other ones. So we decided to go ahead and characterize this Methylobacterium, Agrobacterium, and Variovorax. What we can see here is when we colonize, so this is right after that yellow colonization step, they've all grown together in that super rich medium. Here is our bacillus on its own, and it averages out to 1 like we want it to. And what we can see is that when grown with Agrobacterium, we see a slight increase in how many bacilli on average are attached to the root, whereas with Variovorax, there seems to be a significant increase in how many bacilli are there. What we can do then, though, is transfer it into our maintenance media, and sure enough, we saw that there is a pretty clear difference. This is where it's really important to think about statistics versus trends, and the reason we show all the data. There's a big variance in bacilli on their own. It tightens up a little bit, actually, but we do see an increase in how many bacilli are there compared to bacilli without their helper. And we can go out to that 72-hour time point again, and we see those same trends, that these different helpers are in fact increasing bacillus maintenance on these roots. And one of the questions I get a lot is, okay, well, are these different bacteria just increasing how many bacilli are even in the media? Is this some sort of general growth effect? And what I wanted to quickly show here is that Bacillus subtilis on its own per seedling does increase. These are 2 different days, does increase when we add these different bacteria. So it is something that increases maintenance, but really importantly, it doesn't increase the number of bacilli that are in the liquid medium. So this isn't just a general growth increase, but rather there's something happening where bacilli are attaching to the root more and are surviving on the root longer. Going over to the microscopy related to all of this fun stuff. So in this case, bacilli are going to be false colored yellow. At the top, we can see this overlay of what's called a DIC, but basically it would be what you think of when you look down through a light microscope, like on a bench top. You can see the outline of the root, and although the root has a little bit of autofluorescence, there's no individual bacilli. Compare this to when we colonize with these other helpers. So when we colonize with Agrobacterium and bacilli and look just for the bacilli, we can see these individual spots of bacilli down here, and it's a lot easier to find these in this whole mess of a plant root. And maybe it shows up large enough on your screen, but there's actually so much just gunk around these plant roots, and the bacilli are actually right in the middle of the non-labeled Variovorax. We can then look at the maintenance, which is the step we are really interested in. Again, we don't see those individual bacilli. It's really hard to find them. There's one little guy right here, but if we co-colonize and then do the maintenance step, we can actually still find these individual bacilli along with the roots. And again, Variovorax, there's some little ones tucked into this really big clump of bacteria. And we'll see if these videos work. They're always a little touchy, but yes. What's happening here is this is the bacillus only, and we're coming up through the plane of focus. So this isn't moving, or it's not over time, but rather if you focused, it's sort of like when you're focusing with your camera, if you focus really far away and then you slowly dial back the focus and you can see through this root. And so what you're seeing is that this is the outside of the root, and you can see a few little bacilli, but you can imagine digging through an entire root. It's pretty hard to find these individually. However, if we add Agrobacterium, what we can do is come up through this root, and what we see is a lot more bacilli hanging out, sometimes in these big clumps. And this was pretty cool to us because it gives us an idea that maybe they're interacting with each other, maybe they're touching. Something is definitely happening though, and we can see these helpers really there with bacillus, and it gives us an idea of what's happening when we're looking at those CFU data. And finally, again, we wanted to look at real-world situations, or we wanted to look at real-world situations. You'll notice this doesn't go into soil. That would be another thesis project. But what we wanted to look at was, can we see the same effect on other strains of bacilli, such as FZB42 that was used on lettuce in that picture. And so we went ahead and looked at some of the more commonly used plant growth promoting bacteria that are found in these agricultural different substances, treatments. I chose these as, again, not sponsored or anything, but they do contain FCB42 and GBO3, which are other strains of bacillus, in this case Bacillus amyloliquefaciens, and they have been shown, and these treatments have been shown to help plants grow. We wanted to look at how my helper strains were able to help these bacilli maintain their presence on the root, as well as try to get an idea of, could we mix all of the helper strains together and have them help these different bacilli. So the community idea, if one is good, maybe three is better. Looking at our Bacillus subtilis alone, again, this is Bacillus subtilis on its own versus mixed with our three different other isolates. We did this experiment again, and we saw the numbers that we expected to see. And when we tried the same exact experiment with these two strains of Bacillus amyloliquefaciens, we saw very similar trends. So what this told us, interpreting this graph, is that these helper strains that were able to help Bacillus subtilis were also able to increase maintenance of these more agriculturally relevant bacilli. And like I told you, we wanted to see what happened if we mixed all three of these together and then looked at how many bacilli were on the root. And that's what we did here. This three-member consortium, if you want to compare, basically these are all three together with bacillus and looking at how many bacilli are on the root. And you can see, no matter the bacilli being tested, there is still an increase compared to bacillus on its own, but it doesn't seem to be an increase compared to each of these individual helpers by themselves. And that's pretty much my thesis. So in conclusion, my aim one was to build this new system, and we got it up and running. Building a system was really tricky. I had some great help from some undergrads and had a lot of fun doing it. We were able to validate this system with a different PGPB and with different strains of natural soil-dwelling bacteria. So we got a good idea of, would this work, and could we ask the questions of maintenance that we were really interested in. That aim two was to take this huge collection, 96 strains that were originally taken from the plant Rhizosphere, and see if any of them could help bacillus. What we found along the way is that about half of these would help, or would be able to maintain the colonization on their own, and 22 of them looked interesting in terms of helping bacillus. We then narrowed that down to 3 for further characterization, and we got a better idea of the kinetics, or how this works over time, as well as taking a look at the localization, or where they are on the plant root. And my third aim was to take this and transfer some of this information over to a more agriculturally relevant system using these two strains of Bacillus amyloliquefaciens. What we found there, and just recap, is that These 3 strains in combination don't seem to have much of a different effect, but they do each individually and as a group help these different strains of bacilli maintain their colonization on the root. I want to leave enough time for all of your questions and just to kind of chat about things because this has been really fun. I started in like August of 2014, so I could probably spend about 3 hours doing acknowledgements. And most of the time in public defense, this is the time where you show a bunch of pictures of friends and family. I can't tell you how important my friends and family have been. In 5.5 years, there's been a lot of people who have supported me and really been there for me when I need it. Grad school is a really, really, really hard time. So I do want to thank my parents and my family and friends and partners and social media who have become, real friends and some of my best friends actually. But to sum it up, and this was just a screenshot of my acknowledgements page in my dissertation, my thesis is dedicated to those people, and you know who you are, who can hear someone say, I'm not okay, and their response is, that's okay, how can I help? We all need help in grad school, and the most important thing you can do is to be empathetic and to just try to understand where they are coming from. So thank you to those people who, when I haven't been okay, haven't shunned me, haven't felt bad for me, haven't pitied me, haven't run away from me, but rather have said, that's okay, you're still important, you still matter, I still want to be your friend, is there a way that I can support you? And with that, I think I'm going to flip back over and see if anyone has some questions. We'll see if anyone's still there. I will tell you, this is low-key terrifying. All right, switching back. Okay. Let's see. Do we have some questions? We have our lovely moderator who is taking care about things. So from Phonetical, I have a question of, do you do this without soil, and does the medium matter? This is a really good question. So we did go ahead and not do this in soil. Soil adds even more complexity, and I would love to see some of this work transferred over to a soil or a field situation, and that's really where it would matter in the end. The reason that the medium mattered for us, medium again is the liquid that the bacteria are growing in, that they're getting their nutrients from if they're not getting it from the plant. And in our colonization step, there's a bunch of sugar that's available for the bacteria, and Bacillus really enjoys living in there. The maintenance liquid, however, this is called Maraschigian scourge, whereas our colonization medium is just lab grade LB. In this Maraschigian scourge medium, there's not that many carbon sources. There's not a lot of sugar for the bacteria to use. And so that's why we didn't want to look at maintenance in this media, because for us, it didn't make sense to look at how bacteria were interacting with the root if they had a bunch of sugar available outside of them. These bacteria were then able to, once they were attached, probably get their sugar. The idea is that they might get their sugar from the root that they're attached to in the maintenance. That's a really good question. Thank you, Phonetical. Okay. And again, Phonetical always asks really great questions, is popping into my Ask Me, I'm a Scientist pretty constantly. Phonetical asks, could surface texture play a role? So as a follow-up to the soil actual physical structure could play a big role. Again, we're floating these in liquid, but I'm not sure if it's that or rather if it's the surface texture of plant roots. And yes, the bacteria sort of prefer, it seems like they prefer to go ahead and stick into the little root hairs. I might flip back over to here so that I can pull up one of those slides. As you can see in my pictures, I have to say it's really weird to just be doing this in a silent room. As you can see in some of these pictures, here we go. There are these little things called root hairs. These are super tiny, and the bacteria really like to stick to the surface of the root. They like to stick in these little crevices between the root hair and the root. So the numbers of hairs could have an effect on how the bacteria are sticking. The different regions of the root really matter. This is kind of the upper mid part of this root. So the texture of the root matters, and also how healthy that plant is. They put off different chemicals. All right, let's see. Okay, wow. Y'all have good questions. Emmerich Larkin, did you ever perform marker gene or metagenomic investigations using your hydroponic system? Okay, let's break this down a little bit. So, marker gene or metagenomic investigations, basically, remember that FCB42 graph that I showed you at the start where they looked at the plating of the different bacilli, but they also looked at the genetics directly over here, where selective plating, this is looking for the bacteria the same way that similar that we were doing is breaking them all down and looking at how many different colonies would show up. Versus metagenomic analysis, what they're doing here is they're looking for specific genes. So it's sort of like, rather than, if you wanted to see how many people were in a room, rather than gathering them all up and then counting them individually, and being like, how many of these people have a name that starts with an S? Maybe you're trying to see how many of the people in the room have a name that starts with an S. Instead of grabbing them all out and sorting them by S, you might call, hey, do we have any Sams? Do we have any Susies? Do we have any Sallys? And get a better idea that way. And so identify it by more specific markers. The short version of all of that is no, we didn't go ahead and try that approach because we were only looking at most 4 bacteria at a time. So it was pretty easy for us to look at and pull those individual bacteria out just by the simple plating. It's a little bit faster. However, we've thought about doing this in soil, which would be more complex. There'd be a lot of different bacteria there. Or if we wanted to take maybe all 96 of those isolates, dump them all together and see who sticks together, then we would have to do that sort of survey approach of saying, okay, where is our bacillus? Do we have any variovorax? Can we pull those out individually? Okay. Mark C. says, how related are subtilis and amyloliquefaciens? Would you expect to see similar increases in maintenance in bacilli from the Sirius group? Woof, we got some bacillus people up in here. Okay, so subtilis and amyloliquefaciens are actually pretty close to each other. And in fact, I believe it was GBO3 that was originally identified as Bacillus subtilis. It's actually been, as time has gone further and we've learned a lot more about genetics, we've started separating out these groups more and more and more because we've realized that subtilis and amyloliquefaciens are fairly different. They don't form the same kind of spores. They have totally different amounts of biofilm. So amyloliquefaciens, the 2 strains that I showed you, they form really, really big biofilms on the top of liquid surfaces and respond really strongly to the plant secretions, different chemicals that the plant puts off. But Bacillus subtilis is a lot easier to use. I did at one point go through and look at a huge collection of laboratory bacilli and saw that generally most bacilli did not maintain in this system. Sirius actually, so Bacillus cereus, seemed to maintain a little bit better than the others. So amyloliquefaciens, Licheniformis, a whole bunch, I can send you the list, that they seem to fall off. They didn't maintain very well, but Sirius did a little bit better, not so much though as like the Pseudomonads. Oh, yes, cool. Paul Mathis, hello, nice talk. Oh, thank you. I'm curious if you looked at phenotypes of seedlings. I can imagine that all the gunk, yes, which provides a substrate for increased CFU and mix, also affects nutrient uptake. That was, I wonder if you're one of our reviewers because we just submitted this paper. But no, that was a question we got a lot, is does this colonization affect the plants? And in short, we think it probably could. That's our idea. The problem is in our floating system, we didn't want to end up with our plants in the liquid for too long. So most of our experiments ended after 3 days. I showed one graph where we went up to 96 hours and we probably could go up to 10 days, but just in the short span that we were using, it was hard to tell the difference between the different seedlings. There might have been some difference in the root structure, but it's definitely something that we would love, we as in future people would love to look into. I'd love to know about it. Sorrel Tran, do you think different soil types or areas can affect which types of PGPB are beneficial? I'm sure that the native bacterial populations can competition for nutrients. Yes. Yes, so good. Okay, so we know that bacteria are affected by their interactions with other bacteria, but probably the most important aspect is what is the chemical and physical composition of the soil, right? That it's where the bacteria live, it's where they're taking up everything. And there's a really good question in terms of could different soil types or areas affect which types of beneficial bacteria are actually helping. There's some cool work, I forget who did it, but basically they found some bacteria that were able to help potato seedlings, but then they took these bacteria and put them on the potatoes themselves, and it totally killed the potatoes. So we have to really think about the setting when we're asking these questions about interkingdom, so plants and bacteria interactions. So I think yes, and that was the other reason we wanted to mix all three together and see that yes, all three would eventually come off the root as well, is that maybe one of our helpers would be better in one environment and another could be better, one is better in more of a loamy soil and one is better in more of a swampy soil or something like that. Maybe we could mix all three together and then distribute as a group and still see some beneficial effects. And that the native bacterial populations can cause competition. That's another point, is maybe one of the reasons we're not seeing bacilli stick with the plant very well out in the field is that they're just being outcompeted. There's other bacteria that are trying to take their space. Okay, let's pull up some more. I do have one, though, that my good friend Anthony, where are you, sent to me earlier that I want to make sure to bring up. Of course, he threw me the most hard-hitting question, and I did not read through it beforehand. Okay. So Anthony Burnt said, pretty much every field has some set of differing models or theories of how systems work. These can cause wildly militant factions to form around. In the course of your PhD work, what model or topic in your field did you notice this happen with, and who do you think is most likely right or has the best model? I'm not sure anyone's right. I think that's the cool thing about science, is that nobody's right or nobody will be right for the next 10 years, right? One of the things that I wanted to address with my system, and the reason we switched to this maintenance media, is a lot of the work that is done looking at plant bacterial interactions, if it's done in vitro, so in the lab, in liquid or on agar plates, a lot of that is done with added nutrients. And for bacilli, they don't colonize the plant very well if they are just put into maintenance media. And that's why we wanted to start with a higher concentration of sugars that would promote the colonization of the plants. And what we ended up finding is sure enough, the bacilli could colonize in that way, but they would fall off eventually. So I think that was one of the things that we ran into, was just this idea that everything was fine, I guess. I think that really what we wanted to consider was how much do we want to just believe the data that's out there? How much do we want to take at face value that bacilli are super great, they're sold that way, they help a ton of plants, so just figure out how to put more or how to grow more. Wanted to challenge that idea and say, but are they actually staying that long? What are some of the issues that maybe we're not telling because it's not as catchy? It's not as exciting to say these work pretty well. It's much more exciting to say this is the solution to the entire world's problem. Okay. Wow. Okay. There's so many. Okay. All right. Elena Grossu. Thanks, Susanna. The presentation was really nice. I have two questions. One, what method do you use for establishing CFU? Okay, cool. So what method we use for checking out how many bacteria are surviving on the plant. Again, we take our seedling, we rinse it off, we pop it into some liquid that has some buffers so the bacteria don't just explode. And we sonicate, which breaks up the root into super tiny pieces. We then take that and we do dilutions, because there would, if we found that there's 10 to the 5th bacteria per seedling and we were putting all of that on an agar plate, it would be pretty hard to find that many spots on a plate. So we do a serial dilution, which means that we take that and we dilute it 100 to 1, or we do 10 to 1 depending on our situation. You can think about that like if you had Gatorade and it was super concentrated. Maybe or let's use lemonade. You added a ton of lemonade powder and it's way too strong. You might take like 1 cup of that and 10 cups of water and dilute it down. If you kept doing it, it would be pretty non-dilute. And so we plate across all of those. We also plate on specific agar that only allow bacilli to grow. And that way we don't have to be going through and counting, okay, this is Bacillus subtilis, this is something else. What about Agrobacterium Tumefaciens? So this is the second question of Elena's. What about Agrobacterium? Agrobacterium Tumefaciens is a bacterium that's often used to create GMO because it infects plants really well. And we know it colonizes the plants pretty well. We didn't actually test with Agrobacterium. Part of that is we wanted to use these naturally derived isolates that were coming from the soil. The other part is a lot of people, this is just sort of a straight honesty part, a lot of people are studying Agrobacterium Tumefaciens, and I didn't really want to dip into their zone. It would be something that would be really cool in the future to understand how could this bacterium that's used a lot in the field, how could that have an interaction? Jose Martinez, hi Jose. If you could have a time series of how bacillus behave in different environments, would it be possible to construct a transfer function on how it behaves in different plants and hopefully humans? Okay, right. So this is trying to understand, going back to that longitudinal or over time idea with all of this stuff, could we actually look at how bacillus behaves over time? I looked at that 12, 24, 48 hours on plants in maintenance media. A lot of other groups have done some stuff like this. What you'll see a lot in science papers is that they will go ahead and test their behaviors over time. These time series are really important for us to understand because, again, if you looked at just one of those time points, it would be, you might not get as much information as you need. So I do know that, luckily, Bacillus subtilis, the reason I focused on that one is we know a ton about it, that we have a lot more data about how it behaves in, for instance, like really high salt. We know how it behaves when the temperature gets too hot, and I do think that that is really important for following up on. Zolt Sandor. Do you think this could be applied in microgravity environment like space situation, Mars, etc.? That would be really cool. Astrobiology is awesome, and there's a ton of astromicrobiologists now who are studying how do bacteria behave in space. I really have no idea. I think one of the issues would be for plants, gravitropism. So plants have an idea of where to put their roots. One, they know most plants want to get away from the light. Two, they can actually sense where gravity is coming from. I would be less worried about the bacteria and more worried about the plants in different gravitational settings. That'd be cool though. Jessica Campbell, what was the favorite part of your PhD? Ooh, I like this question. Favorite part of my PhD was definitely conferences. I love talking to different scientists. That's why the favorite part of this presentation is being able to talk with all of you and do this kind of live chat side, even though I can't see you. There's something so exciting about getting together with a huge group of nerds, often in nice locations, and just sharing what you're excited about. A lot of these pieces. So we originally, early on, were looking at Bacillus subtilis and wanted to look in this maintenance system, and we had found it worked with Pseudomonas. We used Bacillus, and we were kind of horrified at how much it fell off. And we were like, other people are saying it stays. What's happening? Like, we would find all these papers that said it stays on there and it's fine. It was really through a lot of conferences and then literature searches and then really thinking about what were the data being presented and what were the conclusions. That's when we really started to think about, oh, maybe this effect is happening in more places, because at these conferences I was able to talk to other scientists in similar fields and just, instead of having that formal back and forth with papers, just be like, I don't know what's going on and I'm freaking out. And they're like, oh, we saw that too. We tried to figure it out, but it's, that's not my main focus. And just having that back and forth was great. I really do empathize with people who aren't able to do that right now. I hope that there's more opportunities for scientists to just chat back and forth. I think that the internet can be good for that, but it's just not the same. If you do get to go to conferences, really push for that. I think in grad school, we're told, oh, you have so many opportunities, and you do in some ways, but you often have to fight your ass off to get them. I luckily had a very supportive advisor, Elizabeth Schenk, in terms of she really promoted us going to conferences. She thought that was important. A lot of times we just have to fight for all of this actually happening. So if you go into grad school and you want to get something out of it, push for it. There's a lot of really hard things with grad school. There's a lot of great things, but the hard things will come no matter what. The great things often require a little bit of bravery and a little bit of stick to it to get to. Miss Luna Dancer, what was your favorite experiment day? I love microscopy. I could do confocal microscopy, which I think makes sense for why this is comfortable for me as well. You set up all your samples, and then you go into this small dark room and you close the door, and there's no one else there and there's no sound, and you just sit and stare through a microscope, and you kind of play with some settings and make it show up as best you can so you can get the best data possible. And there is nothing cooler than setting up something, being really excited about it, and really nervous because you never know. There's so many times that I've gone down to the microscope room, I've set aside entire days or weekends, and it just didn't work. I missed something happened. But when you go down there and you turn on that microscope and you look at your samples, and it's just like, there's the bacteria. There it is. That's where I want to see it. Even if they're blinded, even if that means even if you don't know which sample is which so that you can't be biased, even just seeing those bacteria show up in that bright yellow, it feels amazing. I love microscopy. If anyone wants to show me their microscope, yes, please. Stephanie Hamilton, what's your favorite lab story? Cool. Let's go related to microscopy. I've got a few that I really like, but related to microscopy, a good friend, Braden Tierney, who was at the time an undergrad at Duke, and he's finishing up his PhD at Harvard. We were working on some microscopy together. And at some point in a lab meeting, I said something to the effect of, and this was like a large group meeting, supposed to be professional, and I was trying to give a compliment about how great Braden is, and he's fantastic. And instead of a compliment, I said, if you're going to be stuck in a dark room with somebody, you better be as excited as they are. And I don't think I've ever been more embarrassed more quickly. Now it's funny, and it's also not in front of people. I'm definitely going to regret saying that right now. But I think those sort of things. There's also one of my other favorite things. I'm actually not very good at growing plants. I'm getting better, but it was a joke in lab that I was using plants and I was terrible at growing them. So one of my friends, Michael Yakovidis, he put a plastic plant in one of my containers, and I actually still have that plant in the container, and it kind of just reminds me to not take things so seriously, even when you're doing science. Adriana Carolina Hernandez, Carolina maybe? I know you are focused on bacterial maintenance, but did you see any effects on plant growth during your experiments? Again, that was a really big question, and y'all are right on the money in terms of this is the biggest thing that the next person needs to address, which is, okay, we've done all these things, we have this hypothesis that, okay, now we can get bacillus to stick on the root for longer, but does it matter to the plant? And we don't really know. We, in our timescales, didn't see differences. We know that bacillus on its own has effects on the plants. Even in our hands, some of the stuff I didn't show you is just putting bacillus on a plate with the plant. Even if they're separated down the middle by a piece of plastic, you can still see when the bacillus is there. It produces volatile compounds, so different chemicals that come out into the air, and it changes the shape and structure of the roots. So, but in our exact system with multiple species, we don't know yet. Phonetical. How difficult was it to juggle anomalous data? Very difficult. So like you saw, there is a pretty big variation in some of those samples. And when you're looking at one replicate, so you do one bioreplicate, so on one day you have all your samples, and within a bioreplicate it's pretty small. The averages are pretty similar, and that's really exciting. When you do a second day, you might get a sample set that's here, or you might get a sample set that's down here. And then on the third day, maybe you get it here or you get it here. And so some of the difficulty was not jumping to conclusions. So rather saying, okay, these are the data, these are what we got, let's do it 2 or 3 or 4 times, not 2, but we do it 3 or 4 times, or let's do it one more time. And just being really honest with what we found. One of the things I'm really proud about is that we submitted this paper we did some statistics, which you saw there, but we didn't try to justify too hard. We just said, these are the trends. I hope that you can appreciate them. They're clearly different, but we'd have to do a ton more samples to take those trends and turn them into something that is statistically significant. And I am glad that in this system, which is still very synthetic, that wasn't our main goal. Now, once this gets transferred in the field, and once we would want to address those questions that you all had about effects on the plant, that's where I think it is really, really important that we get to that point where we can easily put on a package, like this is how much it helps, this is for how long it helps, and consumers can be really educated. Phil Rouge, did you investigate any possible metabolites, so hormones, plant flavonoid, etc., helping the attachment to Rhizosphere? We started to. So this is the idea that, again, bacteria are producing all these chemicals. I said at the start that they interact with each other because of this communication. We started to do that. One of the nice things with Bacillus subtilis is that there is an entire library, so there's a whole collection of bacilli that are unable to sense certain metabolites or unable to make them. These other isolates were from the soil and haven't been genetically manipulated, so we haven't made a bunch of mutants or changed around what they're able to do. So it's been more difficult on that end. But one of the things we are interested in, or at least I'm interested in finding out, is if we take these bacilli or we take these other strains and we try to nail down which chemicals are important, or which, maybe it's a biofilm thing. We didn't talk about biofilm, but it's essentially some goopy substance that different types of cells make. And maybe it's the sugars and proteins and things like that that are happening. We really don't have a mechanism for the effects that we were seeing. Stephen Lanton, what caveats would you give for extending research results to bacterial communities on non-Arabidopsis plant or bacteria other than bacilli? So we know that each individual setting is really important. This is one of the difficulties with doing biology, and this is kind of called basic or exploratory biology, is that while there is sort of an applied bent, I have some idea of where I wish this research would go in the future. It's not in a certain way that is directly applicable. So Arabidopsis, like I said, is that kind of lab rat of the plant world. And the findings we have over here they don't necessarily apply to tomato plants. They don't necessarily apply to maize. I think the information is useful. That's how we do a lot of our science, is we pick up little pieces of information in one setting that is what's called tractable, or basically easy to use. We can change it around pretty quickly, and taking it into more complex situations. So if you think about the field, or you think about a plant we don't know as much about the genetics, and we go back to that idea of trends, so many things can change that it's really hard to pin down what was the individual factor that mattered. So this is sort of that reductionist approach where we reduce to as few variables, so as few changing things as we possibly can. But the caveat would be, I think that these data should be represented exactly as they are, which is that this was done on a very specific type of plant. It's exciting for what it is. We hope that it could have importance in these more agriculturally relevant systems. But we don't know that. This is just another building block, hopefully, towards getting to that endpoint of having this be useful agriculturally. Okay, we've got more questions. Try to get to as many of these as I can. Okay, Sorrel Tran, what are your plans now that you're done with grad school? Great job. Thanks to this coronavirus, I don't know. One of the upsides, and I think it's really important to think about the upsides of being stuck at home, is that I've had a lot of time to think about this. I'm going to continue with science communication. Like I said, the most fun thing for me was being at conferences and talking to scientists, and this is really fun to just be able to chat with all of you. So continuing that communication aspect, possibly working more on the biotech pharmaceutical side in terms of science communication for consumers and working with businesses, those sorts of things. And I also am going to be running PhD Balance, which is the support group that the community itself has helped me a lot. And I hope that it's somewhere that students can use different spaces to share their experience and help others get through some of these really difficult times. Zine Lee Sun, do you have any hypothesis of molecular mechanisms about bacillus benefiting from co-colonization? Kind of a similar question as before, but as far as mechanisms, not really. It was super exciting to me to see that the bacilli are actually encased in that big clump of Agrobacterium, and it seems like in general they are touching each other. That could be that they need to be really close for chemical exchange, or it could be something physical where their outsides are touching each other and something is happening there. Quick hypothesis is just that bacilli need to form a biofilm in order to stick onto the plant, and so it's possible that interacting with these other bacilli might be increasing its biofilm production, or maybe they can share some of their biofilm with others. That's kind of the one that is most talked about. Off-trail dino. Do they work in aquaculture? I have no idea. They might. That might be a better application of some of this stuff because it's all been done in liquid. Rapid fire. Phonetical, what's your biggest mistake or your worst day? My worst day was, wow. I mean, I failed my written exam. That was a bad day. But probably my worst day was when I worked with a group of people, and I was leading this group, and I got a review back from somebody. I put out reviews, and like most people said, oh, it was really good. It was running this group. A lot of people said it was good, it was useful, and somebody else just like panned me, and it was vicious. And that was the first time I'd gotten like mean feedback. And that was really hard, but I'm glad that it happened. It happens a lot on the internet, and now it sort of rolls off of me. I've learned certain coping techniques as far as when you see something mean, make sure you're in the right place to take it. You don't have to look at it immediately. You don't have to like blow through it. Just take some time, or even have someone else read it and say like, is this useful? Is it not? Okay. Bernard Offman. Great talk. Aside from your Jove paper, which I could trace back through Google Scholars, any other papers published or in the pipeline? We have submitted one to Phytobiomes Journal. We got positive reviews back, and we're working on resubmitting. We got the reviews back right before our lab shut down, so we're hopefully resubmitting pretty soon. We'll be asking for kind of a decrease in the experiments that were requested, but Phytobiomes has been awesome. We also have a mini review, and there's a bunch of little tail ends of research that could be continued. Yash Akchara, with today's gene editing technology, why can we make a plant so that we can eat from the roots to the fruits and in between? Maybe. I mean, some plants we do. You think of like the broccoli plant. There's so many different pieces of it that we use, and so, in my opinion, it might be better to more variety is usually better, because if the growth system changes, if we have more options of crops, then we can go ahead and actually grow them in different settings. So I wouldn't want us to be completely reliant on one crop. But going back to, I think, Mark C's question of could we use this in, maybe it wasn't Mark C's, could we use this in space? Maybe we would. So I don't know. Plant genetics isn't my focal point, but if someone wants to talk about it with me, let me know. Oh my gosh, there's so many questions. Doug Kurtz, do you have any advice for undergrads interested in environmental microbiology searching for PhD programs? Absolutely. Talk to different scientists in those programs. It's a lot more work, but it's better than any other information you're ever going to get, period. Like, graduation rates are important if you take them seriously. You want to know, is the title of the school, it's sort of important. Don't do it based on like, is this the coolest name? Because if you don't get through it, or if you leave and you hate science, it doesn't matter where you went to. But talking to current students and asking them open-ended questions that aren't yes or no. Don't ask, is this a supportive environment? Ask, how have they dealt with a specific crisis? Ask how they dealt with COVID. Ask how they've dealt with a student having issues with their advisors. Ask the questions directly, because every single department is different, and it's not regulated between. So two people in different labs might have totally different experiences. And for instance, the requirements to get through a PhD in microbiology at UNC are totally different than biology, even though I work in the biology building. Matthew DeForest, what do you wish you could be telling us about but couldn't quite demonstrate? Ooh, that's a great one. Matthew always has great questions. What do I wish I could demonstrate? I do wish I knew more of those mechanisms. I wish I had higher resolution microscopy, because the stuff that I can see on this huge screen, when you can move it around and really find the stuff, it shows up better. But I think it would be cool to understand, how is this actually happening, not just that it is happening. Also be cool, we have something called transparent soil, which has the same refractive index as water, where you pour water into it and it sort of looks like fishbowl pebbles, but you pour in water and you can see right through it. We wanted to get that up and running and start testing in that system, but it was just a pain. So last one in my list, no more questions, moderator. Adriana Carolina, have you set a co-culture of Bacillus subtilis and your helper bacteria on a plate? Have you observed any phenotypes? Oh, you're so good. I think you've asked several questions that my reviewers asked. Yes, and that was the last experiment that we were able to do in time of COVID, was to actually put all these together on plates. And they don't really seem to interact with each other. They don't change each other's growth. They don't change each other's size. Maybe that's the plate situation. Maybe if we put them all together with a plant on a plate. But that was a big question that came back, is maybe these are just interacting in general. And we found that, in fact, at least outside of this system, they were not. Okay, ran a little bit over time. Thank you all for sticking around. This was really cool. I am very open for questions all the time. I'm going to skim back through and see if there's anything that I missed, but feel free to hit me up on Twitter, Instagram, whatever, if you have other questions. And yeah, cheers to almost 6 years of grad school. It was worth it for me. I learned a lot. It was really, really hard. But in the end, I am really proud of this. And it's not something that I would dissuade other people from. If you want other input about how the experience was, I talk about that a lot, but I'm definitely available to ask or answer specific questions. So thank you again so much. I hope that you have a great rest of your Monday. All right, bye.